Hi, I'll be continuing with the history of microbiology. It's the second part of medical microbiology history. So we were talking about Robert Koch. Robert Koch is a German general practitioner. He is known as father of bacteriology. He had demonstrated the presence of or the importance of solid media for culturing of bacteria, where they started using agar as a solidifying agent. So the solid media concept started with Robert Koch. He also introduced methods of isolation of bacteria in pure culture. The hanging drop method for testing the motility of organism also was introduced by Robert Koch. He discovered bacteria like anthrax bacilli, which cause anthracis, and then tubercle bacilli, which cause tuberculosis. He introduced us to the staining techniques which are followed even now. The main contribution of Robert Koch is the Koch postulate. Koch postulate says a microorganism can be accepted as the causative agent of an infectious disease only if 4 criteria are fulfilled. So he had given us some postulates which has to be satisfied to say an organism is the causative agent of a particular infection. So even Koch postulates have some exceptions now. Even now it is considered as one of the important milestone in the history of microbiology. So the Koch postulate says, number 1, the microorganism should be constantly associated with the lesion of the disease. Number 2, it should be possible to isolate the organism in pure culture from the lesion of the disease. Number 3, the same disease must result when the isolated microorganism is inoculated into suitable laboratory animal. Number 4, it should be possible to re-isolate the organism in pure culture from the lesion produced by the experimental animal. So going to the first one, the microorganism should be constantly associated with the lesion of the disease. So if a patient is suffering from an infection, the organism which is causing the infection is supposed to be present in the sample collected or the lesion shown by the patient. So when the patient is suffering from the infection, we can collect the sample which contains the microorganism. That is very helpful in diagnosing the infection. So the first point is that the organism should be constantly associated with the lesion of the disease. The second one is it should be possible to isolate the organism in pure culture from the lesion of the disease. It says that Koch had given us the method of isolating the organism in pure culture media. So the sample can be collected from the patient which is supposed to contain the microorganism and can be grown in the pure culture media in the lab. That will help us in identifying the infection, but at the same time it will help the confirmation that the organism is causing the infection. So it should be possible that all the organisms which are supposed to be the causative agent of a particular infection should be cultured from the lesion from the disease. That is the second one. Third point says that the same disease must result when the isolated organism is inoculated into a suitable laboratory animal. Here what they say is we have an experimental animal model, and if you inject the organism which is isolated in the pure culture medium, if the animal should start showing the similar symptoms which was shown by the patient. So when we are collecting the sample and growing in the lab, after that it should be inoculated into an experimental animal and the animal should start showing the symptom of the same infection which was the patient was suffering. So that is a third postulate. And the fourth postulate says that it should be possible to re-isolate the organism in pure culture from the lesion produced by the experimental animal. Here it says that once the animal shows the symptom, we should be able to re-isolate again the same organism from the patient's that animal to the culture media again in the lab. That is Koch postulate. So we have 4 postulates to be fulfilled to say a microorganism is supposed to be the causative agent of the particular infection. Now there is an addition criteria which was added later, that is the 5th criteria of Koch postulate. So it says that antibody to the causative organism should be demonstrable in patient serum. So this is the latest one which was added later, not by the Robert Koch. So this says now the patient is suffering from an infection, that organism acts as an antigen and the antibody should be present in the serum of the patient. So that is a additional 5th criterion which was added to Koch postulate. Though most of the organisms agree with the Koch postulates, there are few organisms which cannot satisfy the postulates still considered as the causative agent of the infection. For example, Mycobacterium leprae and Treponema pallidum. These two organisms cannot be cultivated in vitro. We cannot cultivate in the artificial medium. And again, we have an exception, Neisseria gonorrhea or gonococci, which has no experimental animal model. So that means we cannot inject this organism into an experimental animal and show or demonstrate the similar symptom which was shown by the patient. Coming to the molecular Koch postulate, this is the modification by Stanley Fatwo in 1988. It's called as model of the molecular Koch postulate. According to him, what it says, a gene of a pathogenic microorganism that contribute to a particular disease should satisfy all the criteria of Koch postulates rather than the microorganism itself. So he says it's not the microorganism, it should be the gene of that particular microorganism which should satisfy all the postulates of Robert Koch. It also says the virulence rate under the study should be associated much more pathogenic strains of species than with the non-pathogenic strains. Inactivation of gene associated with the suspected virulence should substantially decreases pathogenicity. Replacement of mutated gene with the normal wild type gene should fully restore pathogenicity. The gene should be expressed some point during the infection and disease process. Antibody or immune cells directed against the gene should protect the host. That's a molecular Koch postulate. Now coming to the last and the most important criteria, that is Koch phenomenon. According to the Robert Koch phenomenon, guinea pig which is already infected with tubercle bacillus developed a hypersensitivity reaction when injected with tubercle bacilli or its protein. So this connects hypersensitivity and immunity of the of the patient. Coming to the other scientist, we have Paul Ehrlich, who got Nobel Prize in 1908. He was a German scientist. He is known as father of chemotherapy. He discovered sulfurescent arsenical compound, which is now known as magic bullet. It was used against syphilis infection caused by Treponema pallidum. He also discovered the technique of staining tissue or blood cells. He had described the acid-fast nature of tubercle bacilli for the first time. There is an Ehrlich phenomenon which is connecting toxin and antitoxin interaction, and side chain theory of antibody production was described by Paul Ehrlich for the first time. There is an Ehrlich Institute in his name in Germany. Coming to the other Nobel Prize winners in microbiology, in 2005 in physiology or medicine jointly went to William C. Campbell and Satoshi Omura and Yu Tu. Mr. Campbell and Mr. Omura won it for their discovery concerning a novel therapy against infections caused by roundworm parasites, while Ms. Tu won it for his discovery concerning a novel therapy for malaria. The Nobel Prize in Physiology or Medicine in 2018 was awarded jointly to James B. Allison and Tasuku Honcho for the discovery of cancer therapy by inhibition of negative immune regulation. Thank you.